Our last speaker is Xuan Ji on self-sustainable biosolar cells based on bacterial photosynthesis and respiration.
Today I'm going to introduce you a self-sustainable biosolar cell based on bacterial photosynthesis and respiration. And concerns about global climate change and fossil fuel depletion has been increasing in recent years. Several alternative energy sources have been proposed to solve this issue, including hydrogen and ethanol fuels derived from biomass. However, these energy sources are all secondary sources that that involve the step of storage and transportation, and also involve safety issue concerns. So we want to ask ourselves whether we can use the primary source, the solar power, directly for power generation. We already have the semiconductor-based silicon solar cell on the market, and its working principle is in close analog with the natural photosynthesis. However, the conventional commercialized solar cell can only work at daytime. At night, when the sunlight, it can no longer work. So can we create a hybrid system by incorporating the natural photosystem into the artificial solid-state system to work even when the light source is not available, say whether it can work at night or in cloudy and rainy days? Here is the working principle of the system. When light is available, electrons are extracted from water and delivered to the electrode, or they can be used to reduce carbon dioxide to synthesize carbohydrates. When light is not available, say in dark condition, carbohydrates are decomposed and electrons are released through the red arrows, and this way the power generation can be sustained at both night and day. And here are the primary reactions involved in the system. As you can see here, electron originated from water will finally be reincorporated into water. And in this manner, electron can be recycled in the system. So water serves as the only electron source. And also it has a large intermediate chemical step in the form of carbohydrates. And is this feature that allowed us for sustainable power generation at both night and day and night. You may then want to ask, what is this process good for bacterial? Or how does bacterial gain the energy for its own reproduction in the process? So as can be seen in the movie, along with electron transport, protons are co-transported to build up the proton gradient along the membrane. And this electropotential gradient can later be used to drive ATP synthesis. And this way, bacteria can maintain its own life. And in this way, those microorganisms can be envisioned as micro workers in the system for electricity generation. They simply drive the electrons from a lower potential to higher potential for power generation, and meanwhile they can synthesize ATP for their own reproduction. And here we demonstrate a paradigm of synthetic biology system in a broader horizon. I think this is particularly significant for large-scale applications. In addition to functionalize the biological parts, we need also to consider the surface chemistry and the physics to fabricate a seamless system. And bacteria in this system are confined to drive electricity generation. Meanwhile, they are eating up carbon dioxide. Therefore, the system has a dual function. It can function as a power generator, or it can simultaneously sequester carbon dioxide. And here is our preliminary results. In this first 4 days, we continuously illuminate on the system. And you can see both the power output and the open circuit potential remain steady during this illumination period. And after that, we moved the system into a dark box for 62 days, or 2 months. And during the prolonged dark period, the power generation can still be sustained. And this energy actually is stored in this period. So you can see the efficiency is quite high. And even after the 2 months dark period energy generation, we illuminate again onto the system. The power can still be regenerated. It means that the biological compartment here does not lost their adaptivity from the photoautotrophy to chemoautotrophies. The adaptivity is not lost during the prolonged process. And we then want to know whether the system can operate in real situations. So we operate in 12-hour light and dark cycles to mimic the diurnal cycles. You can see the recoverability of the power generation is quite good. And also another feature is we use the first order exponential decaying function to feed the curve, and we found that actually you can see the power decrease much slower than its increase. It means that if you consider it as a battery, it can charge up much faster than its discharge. And we then examine the property of carbon dioxide sequestration. We find that the carbohydrate synthesized and stored inside the cell and also secreted into the solution. And the total carbohydrate synthesis rate under illumination is about 16 times larger than its degradation rate in the dark. And this property guarantees that when this system is operated in 12-hour dark and light cycles, the net emission is negative. So this property is quite unusual because the best result for conventional fuel cell or other microbial fuel cells, you can only achieve carbon neutral. But here we can do carbon negative. And we further evaluate whether the operation of carbon dioxide sequestration will interfere with power generation. So we replace the culture medium. You can see in this period, the system is immersed in the culture medium, which already contain a high level of carbohydrates. We then replace it with fresh medium that does not contain any organic carbon. And after the replacement, in both light and dark conditions, the power can still restore to its original level. And this result suggests that the two functions are compatible to each other, and it can truly function simultaneously for power generation and carbon dioxide sequestration. And here is our current energy scheme. The carbon dioxide emitted from the combustion machine by burning the fossil fuel can now be reincorporated into the fossil fuels and left a gap in the circle. And this broken circle does not allow sustainable energy. But here we hope that with the power of synthetic biology and synthetic biology, we can fill up the gap because our system is carbon negative. And with this closed circle, we can transform our economy towards a carbon neutral, balanced, sustainable energy. Okay, thank you very much for your attention. Please feel free to ask me questions.
Thank you. Are there any questions? Yes, Chris.
Okay.
Hi. So when I first heard about microbial fuel cells, I had a very hard time understanding them until I saw a presentation by the Navy where they're a very viable route to having sensors out in the ocean that can perform forever. I was wondering, for your system, what is the first application that you're imagining where this could actually be utilized?
You're talking about applications. Then I should mention the magnitude of power. Right now, all kinds of biological-driven fuel cells are lower. The power density is lower than its counterpart in fuel cell. But the magnitude achieved in this study is actually comparable. This magnitude is comparable with other microbial fuel cells. And for other fuel cells, you're talking about the remote controlled, the power supply for remote controlled sensors for marine, to monitor the marine environment, right? And I know the paper you're talking about. Our magnitude is comparable to theirs. Yeah. We will also use natural organisms here.
Oh, sorry. What do we do?
I had a question. Yeah, what organisms attach to the anode?
Okay. I'm sorry that because this work
What
has
organism is attached to the anode?
Yeah. Because this piece of work has not been published yet, so I'm not able to disclose the detail at this moment.
I'm not sure how I feel about that. Does the audience want to vote?
But I shall say.
Can you give us a hint?
But I shall say it's not limited to a particular class of strains. I shall say the capability is actually not that unusual.
So my question goes into the same direction. I'm having a hard time to understand what actually is really going on.
Right.
So the scheme is clear, but then how do you have your solar cells immersed in a culture medium, or how actually looks your...
The solution is contained inside the system. The system is not immersed in the solution.
So have you got some scheme that you could show us how it actually looks?
The scheme is like, basically the system look at this. It has the two chambers, and this two chamber construction is identical to that of fuel cell or microbial fuel cell.
And where's the solar cell in that one?
The biological element is in the anode chamber.
Ah, no, I get it. Okay.
Okay.
No, yeah.
Are there any other questions? Okay, thank you very much.
Okay, thank you.